goal of breeding crops, whether through genetic modification or conventional methods, is to produce crops with improved characteristics. All methods result in changes in the genetic makeup. Some examples of conventional breeding are selection, hybridization, polyploidy, and induced mutation. Here are the differences between GM methods and conventional breeding. The basic difference is most conventional breeding relies on genetic variations within the same species, whereas in GM technology, genes can be inserted into crops from unrelated organisms, for example, bacteria to crops. Selection. This is the most ancient method. Plants are selected from a large population, grown and observed. Those with desirable traits are selected and others are eliminated. The selected plants are grown over several years under different environmental conditions for making further eliminations. Finally, the best plants and pure lines are selected as they continue to inbreed among themselves. Almost all our crops are a result of selection. Hybridization. This is when a breeder or farmer observes a good trait in a plant, but also has a poor quality. For example, sweet fruit, but susceptible to bacteria attack. And another crop is resistant to the bacteria. Both the crops are self-pollinated year after year, and the seeds are resown. When identical crops appear, they are known as pure line. The two pure line crops are now cross-pollinated by hand. This results in first generation or F1 hybrid. The seeds of the F1 are planted and they should have the combined traits of the two parents. For example, most commercial crop varieties such as corn, wheat, cotton or rice. Polyploidy. Most plants have two sets of the same chromosome. They are called diploid. Those with 3 or more sets of chromosomes are also common and are referred to as polyploids. To increase chromosome sets per cell, a chemical called colchicine is used that results in doubling of the chromosome number. This results in increase in size, but often have low fertility and grow more slowly. For example, seedless varieties of fruits. Mutation. Mutation is induced by exposing plants to X-rays, gamma rays, protons, neutrons, alpha particles and beta particles, and chemicals sodium azide and ethyl methane sulfonate. This is usually done when genetic variations are limited within the same species. Both the irradiation and chemicals result in genetic changes, and when useful mutations occur, the crops are selected. For example, wheat, barley, rice, potatoes, soybeans, oat, grapefruit and onions. Genetic modification. GM is a technology that involves inserting DNA into the genetic sequence of a crop. To produce a GM plant, new DNA is transferred into plant cells to confer a useful trait. The cells are then grown in tissue culture where they develop into plants and later back-crossed to produce pure lines. For example, insect-resistant corn, herbicide-tolerant cotton, and rice enriched with provitamin A. Now, let's separate conventional methods from methods of modern biotechnology. Conventional methods include selection, hybridization, polyploidy, mutation. Modern biotechnology. Methods include genetic modification, protoplast fusion. Limited to exchanges between the same or very closely related species. Allows the direct transfer of one or just a few genes between unrelated organisms. Little or no guarantee of any particular gene combination from the million crosses or hybridizations generated. Desired trait can be achieved in a shorter time compared to conventional breeding. Undesirable genes can be transferred along with desirable genes. Only desired genes are transferred. takes a long time to achieve desired trait, provides the result in a shorter time. Not regulated. Once the desired crop is developed, it can be released to the market or cultivated without prior approval. Highly regulated to ensure its safety to humans, animals and the environment. And the time taken for regulatory process is lengthy. Once the desired crop is developed, it takes a long time before they are approved for cultivation.
For more information on biosafety in Sri Lanka, visit biosafetyclearinghouse.net.